Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 for October 2021. This is the part 2 video of this paper. I will put the part 1 below the description box of this video. Let's begin. Question 5 says, antimicrobial substances can be tested using a minimum inhibitory concentration MIC assay. This assay determines the lowest concentration of an antimicrobial substance that prevents visible growth of bacteria. So here they're going to say a microdilution plate is used in these assays. It is made of plastic and contains small wells that the antimicrobial substance and the bacteria can be added to. The diagram shows a microdilution plate, so it looks like this, and these are the wells. You can see them inside. That is where the substance is going to be deposited, as well as the bacteria, to check how effective it is. Let's move on. An investigation tested 8 antimicrobial substances on one type of bacteria, E. coli. Appropriate controls were included in this investigation. The diagram shows the steps involved. So I'll look at step 1. It says each antimicrobial substance was added to the wells in one row of the microdilution plate using a 1:2 dilution plate method. Step 2, the same concentration and volume over suspension of E. coli was added to each well. And then step 3, the microdilution plate was incubated at an appropriate temperature for E. coli for 72 hours. This step here ensures that bacteria can grow if it's present. So if the antimicrobial substance is not effective enough, then the bacteria will grow within this step. And then step 4, the microdilution plate was looked at under a microscope to determine which of the wells had E. coli growing in them and which ones did not. So meaning the wells that had E. coli, it means the antimicrobial concentration was not that effective. Those that did not have E. coli growing in them, the antimicrobial substance would be effective at that concentration. Let's move on. So here the first question says, all steps in this investigation had to be carried out using aseptic technique. State the meaning of the term aseptic technique. Aseptic techniques are methods used to prevent contamination of culture with other microorganisms. Usually these techniques are used if unwanted microorganisms are to be prevented from being part of an experiment or part of a process that is being carried out. So that is to be aseptic or to use aseptic techniques. The next part says, describe 2 aseptic techniques that could be used in this investigation. Number 1, we could carry out the experiment besides a Bunsen burner or a Bunsen flame. This ensures that any microorganisms that could be present in the air or around the experiment will be completely destroyed. Or we can use sterilized equipment. The beakers, the test tubes, and everything you're going to use in the experiment has to be sterilized. Another thing you can do, you wash hands and wear gloves. Of course, gloves are going to be sterile, so you wash your hands and then wear gloves. That is okay. We can also minimize the time that the cultures are exposed to the air. Remember, in the air, there are some microorganisms, so if you do not expose your cultures to the air for longer periods of time, you minimize the number of unwanted microorganisms that could enter into your intended culture. So that is creating aseptic techniques. Next, they say, explain why using aseptic techniques in this investigation is important. The importance of using aseptic techniques, here we can see it will prevent entry of bacteria that may compete for space and nutrients with the E. coli. Remember, we are studying how effective the antimicrobial agent is against the growth of E. coli. So it means if other microorganisms are also present, they will compete for space. The E. coli may not grow properly because other microorganisms are growing, and we might think that the E. coli did not grow because the antimicrobial substance is being effective, when that's not the reason. Another part, it prevents the person working with the bacteria from getting infection. This could be if the other microorganism, let me use a smaller one. If the other microorganism you're working with is pathogenic, so this is for pathogenic microorganisms that could be present in the air, pathogenic microbes. So using aseptic techniques allow or prevents the growth of pathogenic microorganisms, which prevents the person working with the experiment or carrying out the experiment from being infected by pathogenic microorganisms. Let's move on. Here they say, explain why the microdilution plate had to be incubated at an appropriate temperature for E. coli for 72 hours. When they say appropriate, it means to allow for E. coli to grow. So I say the appropriate temperature is needed for proper enzyme activity. Remember, any biological process is usually controlled by enzymes. So the temperature has to be optimum or appropriate for the enzymes to work in order for the growth to occur or for proper metabolism within the microorganisms to occur so that they can grow and increase in number. The other part, incubation for 72 hours enables bacterial growth to be visible for the antimicrobial substance to have an effect. So if we just culture maybe for 5 hours, we could conclude that the bacteria did not grow, but actually due to the maybe the antimicrobial substance, but actually it could be because we did not give it enough time for the growth to be visible or seen by our own naked eye. So 72 hours is appropriate enough to allow for the cultures or the colonies to be visibly seen in order to make a conclusive conclusion based on what we have observed. The next part says, the diagram shows the result of the MIG AC from the investigation. So this is how they carried out the experiment. They say, you can see these were positioned from 1 to 12 and this side A to H. So this is increasing in 1 in 2 dilutions of antimicrobial substance. They're diluting further and further. And the last column here, we can see it's a column with no antimicrobial substance added. Then this side we see row A to G used to test each of the 7 antimicrobial substances. You can see from here to here, these are antimicrobial agents. And row H is used for an antimicrobial substance that affects E. coli only. It means if it affects E. coli only, its increase in concentration will prevent E. coli from growing as well. So let's go to the next part of the questions. Please remember to visualize this because most questions are going to be about this part here. So let's move on. So Roman 1 says, explain why an antimicrobial substance that affects E. coli only was included in the AC. In this AC. So we can see row H. Let me take you back slowly. We can see this is row H here. Remember, this is the antimicrobial substance that affects E. coli only. You can see there is going to be a dilution factor. As more dilution occurs, we can see there is going to be more E. coli growth. So we expect the higher concentration of the antimicrobial to be around this area. And as dilution occurs, there is less and less concentration of the antimicrobial agent, and we expect more E. coli to grow as dilution goes on further. So I'm going to write this. The reason as to why we use an antimicrobial agent that is effective only or that affects E. coli only is because we want to ensure that in this case we see since the substance only affects E. coli, only E. coli will die and any other present microorganisms will not die. So we'll be able to know if there was a contamination or not. So here, if they would die, and all the other contaminating bacteria would not, so we will be able to see those. So it would be known if the sample was contaminated or not. Again, one more time, the antimicrobial agent only affecting E. coli, it means all the E. coli will die, and therefore any growth that will be visible on the Petri dish or anywhere you're trying to culture, that would be due to the contamination that would have been caused by any other foreign microorganisms. The next part says, explain why there was one column that had no antimicrobial substance added to it. I want to take you back to column 12. This is the column here. There was no antimicrobial agent added. This is done to ensure that growth occurs at all the different antimicrobials that are being used. So in this case, we had to ensure that growth is going to occur. So working without an antimicrobial agent will ensure that growth occurs so that we can conclude that the places or the experiments that growth did not occur, it was due to the antimicrobial agent. So let me go back. So here I said, to show that the incubated bacteria were viable, meaning able to grow. So if no growth occurred, it will not be known if the bacteria just did not grow or the antimicrobial was the one that affected their growth. So this is to make it more conclusive that growth did not occur due to the antimicrobial only, but not due to maybe the other conditions but only the concentration of the antimicrobial that is being used. The next part says, the MIC for the antimicrobial substance used in row E was in column 4. Describe how a 1:2 dilution plating method would have been carried out to achieve the dilution in this well. So what we would have done is by adding equal volume of the antimicrobial substance and the bacterial growth media together, and this can be done by repeating the culture using the previous solution. So if you had a previous solution here, you take that, put it in here, half, and then add, if it's 1 ml, and then you add 1 ml like that. So this is going to be a 1:2 dilution, something like that. That will be a 1:2 dilution. If you take 1 ml from here and 1 ml from the diluting substance, then we're going to create a 1:2 dilution. So using the previous culture and then adding the antimicrobial agent, like we have said. Going to the next part. Here they say, calculate how many times more effective the antimicrobial substance used in row G is than the antimicrobial substance used in row E. So if we can look at the antimicrobial substance in row E, we can see it is a 1:8 dilution because they diluted and diluted and diluted. So 1:8. Then the one in G was 1:56 because they diluted a 1 basically in 8 other halving times or dilution times, 8 times. So that is the same as 1 × power 8, which is 1:56. So if we have a 1:8, let me call it like that, 1:8 and a 1:56, the difference is going to be a 2 power 5, which is a 32, the difference between the two. So the antimicrobial G, here when they ask how many times, if more effective the antimicrobial substance used in row G is than the antimicrobial substance used in row E, it is 32 times more effective. In this case, we can see that because again, they were asking how many times more effective? 32 times. So E is less effective than G by 32 times, or you would say G is more effective than E 32 times. Question 6. Seaweeds are a group of organisms that carry out photosynthesis. Identifying the types and proportions of chlorophyll pigments is important in the classification of seaweeds and biodiversity studies. State the meaning of the term biodiversity. Biodiversity is the different number of species and the genetic diversity or differences within each of the species. So if we have the more the number of species and the more the genetic differences in each of the different types of the species leads a greater biodiversity. The next part B says, explain the role of chlorophyll in the light-dependent reactions of photosynthesis. Remember, these are light-dependent reactions, and we know that chlorophyll absorbs light energy. The light is going to be absorbed by chlorophyll, and the electrons are going to be released that are used in the synthesis of ATP as well as reduced NADP in order to be taken into the light-independent reactions for the formation of other products that are going to be used in the formation of glucose and so on. So it absorbs light energy, and the electrons released are going to be used to synthesize ATP and form reduced NADP. Let's move on. The next part says, an investigation compared the effectiveness of two solvents, Propanone and Dmso, in extracting photosynthetic pigments. 4 species of seaweed were collected from the Indian coast of Tamil Nadu and taken to the lab where the species were identified. Each species of seaweed was split into 2 samples of equal mass. The photosynthetic pigments were extracted from each sample using one of the 2 solvents and their concentrations determined. So they used either propanone or Dmso in the extraction of the pigments. Let's move on. So here we can see, they say the graph shows some of the results of this investigation. We can see the data for propanone and the data for using Dmso. Here we see for species P, we see that is for propanone, Dmso. Here we see propanone, Dmso, and so on. So the vertical axis is chlorophyll A concentration. So this is for chlorophyll A. And down here we can see the vertical axis has chlorophyll B. The concentration is in microgram per centimeter cube. And here we see the different species that were extracted. And looking down here, we can see the species P Species Q, R, and S, again, this is total concentration of chlorophyll. I would like to show you the whole page here in just one slide if we can. Okay, let me make it smaller like that so that it can be visualized. So here we can see propanone extracts a lot of species P. You can see the concentration of species P in chlorophyll A is in a higher concentration. It extracted more of it. However, we can see for species Q, we can see also propanone did a higher extraction in comparison to what is in chlorophyll A. So here we see propanone extracts more of species Q from chlorophyll B than in chlorophyll A. However, we can see here propanone does more for species R in chlorophyll A than species R in chlorophyll B. So we can see from the data we have differences based on the solvent that is used. We could extract a specific concentration of the pigment within chlorophyll A or chlorophyll B. And finally, from this graph, we can see that the total concentration of species B extracted is actually higher using propanone in comparison to using Dmso. Here, the same thing, propanone than Dmso, but here we can also see the same thing. So overall, we can see there is a higher number of these pigments or these species that is extracted using propanone in comparison to Dmso. So I will go to the next page to look at the questions that are being asked. So let me make it bigger here. So they want us to discuss the effectiveness of these extraction methods in the identification of these species, and they want us to use the information in the graphs to support the answer. So for me, I began by looking at propanone. I said propanone extracts more chlorophyll A from species P and R. This is referring to the graphs. Dmso extracts more chlorophyll A from species Q than propanone. So Dmso is better than propanone. And then we can see that propanone extracts more chlorophyll B from species P and Q. Then also when you look at the graph, you can see propanone extracts more total chlorophyll than Dmso. This part here is for all. I want to take you back. Look at propanone. For total chlorophyll, propanone, we can see it extracts out more total chlorophyll in all species than Dmso did. So you can see this Dmso, this is less than that is less than that is less than that, and that is less than that. So meaning the total chlorophyll extracted from all species using propanone is higher than that extracted using Dmso. So the point I made here is referring to the final graph we saw on the previous page. So the next part I say, therefore, propanone is the most effective solvent at extracting chlorophyll than Dmso. Next I say species B seems to contain the most chlorophyll when using propanone. Let me take this back. And then since total chlorophyll seems to be less than the sum of the components chlorophyll molecules, the next part we see the choice of the solvent used to extract the chlorophyll should greatly depend on the solubility of the pigments in the solvent. This is based on your knowledge. You should know that if you're using solubility as a method of extracting something, whatever pigment you're trying to extract should be soluble in the substance you're trying to use. So if you have propanone and Dmso, and we see that propanone is extracting more of this different pigments, it can tell us that maybe the components are more soluble in propanone than in Dmso. That is why we see a higher concentration when you use propanone than when you use Dmso. So here I say, therefore, more than one solvent, or if more than one solvents are used to extract the pigments to identify the species, so using two or more based on which one is more soluble in which leads to a higher concentration. Also, the method seems effective because it can be done on site, meaning in the field. However, the data does not show validity, i.e. there is no error bars. So there is no changes of variability because there are no error bars. There is no standard deviation to prove reliability of the results. This is my own opinion, most of the data I have observed. If I take you back, you can see it's just a graph. We do not have the error bars to show us which was the minimum, which was maximum, so that we can know that the experiments were repeated and the mean was calculated. Based on the data given to us, we do not know if a mean was calculated because there are no error bars. We do not have the standard deviation. So this could be a good comment on the results, explaining to the examiner that maybe the results are not that valid. Moving on to the next page. Finally, they say suggest why different concentrations of chlorophyll were obtained using these two different solvents. I say, because the chlorophyll pigments have different solubilities, this is very important in the solvent, so we will obtain different concentrations when we use different solvents. The next part, the different solvents used disrupt the cell membrane. Remember, these solvents, you're trying to extract pigments from some cells, so the cell membranes have to be disrupted or broken apart so that these pigments can leak out. So the different solvents used disrupt the cell membranes differently. They do the breaking apart or creating pores within the cell membrane differently so that the contents or the pigments can leak out. So it's going to lead to a different concentration. And lastly, because the pigments to be extracted have different structural arrangements, that could also lead to different concentrations being obtained. This brings us to the end of question 6. I will move on to question 7. Question 7. Climate change is dependent on the balance of carbon released into the atmosphere and carbon removed from the atmosphere. Some ecosystems are better than others at storing carbon in their soil, releasing less back to the atmosphere, into the atmosphere. Conserving these ecosystems could be an important way of reducing climate change. One study investigated the extent of decomposition in different ecosystems. The study used tea in tea bags as the source of organic matter for decomposition. The photograph shows the tea bag. So we can see this is the tea bag, and a sample of a tea bag used in the experiment. Let's continue. So here they say, the first part of the study was carried out in the laboratory. 2 types of tea were used. There is green tea and rooibos tea. A number of unused tea bags containing each type of tea were buried in soil at 2 different temperatures. We have 15 degrees and 25 degrees. The mass of tea in each tea bag had been determined before they were buried. At regular intervals, tea bags were dug up, dried, and the remaining tea reweighed. The diagram shows the results of this study. So we can see the different types of teas. We can see this is for green tea and the other green tea at different temperatures. We can see basically the relative mass of tea remaining is going to be lower, we can see, for green teas in comparison to rooibos tea. And we can see there is greater decomposition at a higher temperature because we can see the relative mass of tea remaining is going to be less for both different types of tea depending on the temperature. So, okay, let's continue down here. They say the tea bags had to be dried before weighing to remove water. 1 tea bag contained 28 grams of tea when it was buried. When it was dug up, it had a weight mass of 42 grams. The water content of the tea bag was calculated to be 50%. Calculate the mass of organic matter lost during this study. So here we can see, because remember they said 42 grams, so 42 grams, if 50%, we can see this is going to be 50% is water. So then 21 grams of water and 21 grams of tea were left. Remember we have 42, and they said the tea bag weighed 50%. So we can see if this is going to be water, it means 50-50. So of the 42 grams, we have 21 grams and then 21 grams for each. So when we continue, I said, so the mass lost during the burial would have been the 28 grams. Remember, it contained 28 grams minus the 21 grams. So this, in this case, is going to be 7 grams lost. And again, I want to go back. Remember here they said the water content of this tea bag was calculated to be 50%. This means during the drying, after the tea bag was dug up, they dried, and 50% of the mass that was present, remember that 42 grams, 50% of this was water. 50% of 42 is 21, so it means 21 grams was water. So if 21 grams is water, yet the whole sample was 42, it means 21 grams of tea is going to be left, yet 28 grams were buried. So to get the mass that has decomposed is 28 minus 21, giving us 7 grams. So this should be the answer. Let's move on. Here they say, calculate the rate at which the relative mass of the green tea decreases in the tea bag buried at 15 degrees Celsius at day 30. So here, let me take you back slowly. Here we will come at day 30. You can see here. Remember, we are looking at the green tea bag at 15 degrees. So you will draw a tangent at this point here and then find the gradient of that tangent. So the gradient of that tangent is going to give us the results. As you can see here, I said using the graph to find the gradient at 30 days and 15 degrees. Using these values, I found it to be negative 0.0077 gram per day. Now, I do not need to use the negative because the negative just shows there is a decrease. So the answer here is 0.0077 grams per day. The next part says, explain the difference in the decrease in relative mass of green tea in tea bags buried at 15 and at 25. Remember here we are explaining the difference, so we can begin by talking about the difference that exists and then give a reason why the difference is the way it is. They want us to use the information in the graph to support the answer. So I said there was more decomposition for the tea bags buried at 25 degrees, of course, than that at 15. Remember, this is because, and again, I want to emphasize every time they tell you to explain the difference, start by writing down what the difference is and give a reason for that difference. The difference I see here is more decomposition at a higher temperature, which is 25 degrees. The reason for that is these enzyme-controlled reactions. So I said because the enzymes that carry out decomposition worked faster at a higher temperature, because when the temperature is higher, there are more frequent collisions that occur between the enzymes and the substrate at 25 degrees than at 15 degrees. And finally, loss in mass is due to the formation of carbon dioxide and water during the respiration of the decomposers like bacteria and fungi. This was an explanation. Remember, bacteria and fungi are organisms, so they need enzymes, and they have enzymes within them to help them with their biological processes, and those enzymes work at certain temperatures. 25 degrees is higher than 15, which is good or more adequate for enzyme activity, leading to more collisions between the substrate and the enzyme in order for the process to occur faster. Let's continue. So here they say, suggest why the rate of decomposition of green tea was different from that of rooibos tea. Now, these are different types of teas, and they could have different molecules within them that can be decomposed at different rates. So I say the different teas may contain different molecules that cannot be easily accessed by the enzymes at the same time. So that could lead to the difference in decomposition. Next one I said, they say, in the second part of the study, several tea bags were buried in different ecosystems. The tea bags were left buried for 3 months and then dug up. A number of measurements were taken from the tea in these tea bags, and the mean decomposition rate of the mean stabilization factor S calculated. The higher the value of S, the more carbon is stored in the ecosystem. The graph shows some of the results of this study. Now we can see these results, or this data shows us their error bars. You can see vertical and horizontal. So these results were carried out at multiple times, or they were repeated. A mean was calculated, and we can see the standard deviation, so they can be more believable. Let's move to the next part. Here they will ask, suggest why each point on the graph has both a horizontal and a vertical error bar plotted with it. Vertical and horizontal error bars are going to tell you that the experiment was carried out multiple times, and then a mean was calculated. So I said because the values used in the plot for stabilization factor S and decomposition rate are both mean values. Because they are mean values, there is going to be some error bars. So because there is a higher value, a lower value, and then the mean that was used. Lastly, here they say, explain which ecosystem should be conserved to have the greatest impact on climate change, and use the information in the graph to support your answer. And when I go back to the graph, and so that we can see, since this is decomposition, right, and the horizontal value is stabilization factor, I am going to go with this, number 6. Again, remember they said the number of measurements were taken from the tea in these tea bags, and the mean decomposition rate and stabilization factor S. We want to know which sample is going to cause. Remember, this experiment is looking at, I want to go back here. They say the tea bags were buried for 3 months and then dug up. A number of measurements were carried. We are trying to find how much of these substances are contained within a specific ecosystem. So I'm going to try to explain why I chose number 6. I chose number 6 because it has the highest stabilization factor, so more carbon is stored in the ecosystem. Remember, the purpose of this experiment is to know which ecosystem is going to store more carbon, not release more, but store more. It means decomposition is going to be slower. So I said it also has the lowest decomposition rate. If it has lower decomposition, it means most of the CO2 is going to be, there is no release of CO2. It's going to be contained within the sample, or it has a slower decomposition rate. So the microorganisms that are trying to decompose it are acting really, really slowly. So there is going to be less CO2 released to the environment, which is good for climate change. So most CO2 will be retained as stored carbon and not released to the atmosphere. And then there will be less contribution towards global warming and climate change. Again, to take you back here, we can see this part here, since the vertical axis is decomposition, it had the least decomposition, yet it has the highest stabilization factor, so we can know that this is going to have the least release of CO2 to the environment, leading to the least change in causing climate change. This brings us to the end of question 7. I will move on to question 8. Question 8. Scientists study the distribution of biomass in organisms on Earth. Say the meaning of the term biomass. Biomass is the amount of organic matter present in an organism. That is what you should write. Part B says Voronoi diagrams were used to represent some of the data. 2 Voronoi diagrams are shown. There is diagram A, which shows the biomasses of groups of organisms, and diagram B, which shows the biomasses of organisms in the animal group. The biomass is given in gigaton of carbon, GTC, where 1 GTC is equal to 10 power 15 grams of carbon. So when we look here, we can see there is diagram A, whereby the plants have the highest biomass, and then we go to the bacteria, and then the others follow. Then we have diagram B, where we have the arthropods having the greatest biomass, and the rest follow. One thing we need to know about the Voronoi diagrams is it's easy to observe which one is actually the greatest. I can observe here and see plants have the greatest biomass, and here arthropods have the greatest biomass, and then the fish. However, for something like this one and this one here, because they have a shape, they have different shapes, even if they have the same biomass, it could be hard for me to know that due to the difference in their shapes, so I could misinterpret the data. The same thing occurs with mollusks and annelids. Here we can see it's still 0.2 GTC, 0.2 GTC, but you could be misinformed about the number of organisms here if you do not properly calculate the area contained in there. So let's see. They're going to say the area of each polygon is proportional to the biomass of that organism. The shape of each polygon has no meaning. So we should know the shape has no meaning. All we're interested in is the area because it gives a correct representation of the biomass contained in that region. The first part, they say calculate the percentage of biomass in organisms belonging to that domain Eukarya. They want us to use information from diagram A. I want to take you back to diagram A so that we can look at which ones are the Eukarya. Diagram A, we know eukaryotes, the plants. We know animals are Eukarya, fungi are Eukarya, and protists are Eukarya. So I'm going to add that, and that to get the total Eukarya. When I added the total Eukarya, I got 468. But remember, this question is asking us to calculate the percentage of biomass in organisms belonging to Eukarya. So it means Eukarya over the total number of organisms. So total organisms should be the archaea, bacteria, and viruses plus the Eukarya. When I added everything up, I got 545.2. So the percentage is going to be that divided by that times 100, which gives me 85.84 or 85.8%. And that should be your answer. The next part suggests why the scientists study the distribution of biomass in groups of organisms and not the number of individual organisms. The number of individual organisms would be really hard because you have to count the different numbers, and some animals are going to be so many. Another thing is some animals have different sizes, so it could be hard to categorize them in number. However, biomass puts everything as a group, which is easier for somebody to understand. So I said because they could not count all organisms as individuals, that would take too long. And also organisms in a certain group can have different sizes and therefore different biomass. That could be misleading if you counted them as numbers. But when you put them as a group, it's easier for you to interpret. The next part says, discuss the advantages and disadvantages of presenting data in Voronoi diagrams. and use diagram B to support your answer. The advantages are Voronoi diagrams are easy. They're an easy way of representing data, and they can easily be understood because it's easy to see the area, and you can know one area is bigger while the other is smaller. The example is, it is easier to observe that arthropods have the greatest biomass. Let me take you back to diagram B. We can see here, by looking at this Voronoi diagram, we can see that arthropods have the greatest mass, and then this comes next. The problem is looking at this and that have the same biomass, but they are different shapes, so that could be misleading and confusing. You have to be able to understand it fully or properly. Another one is these mollusks and annelids. They have the same biomass, but their shapes are different, and that could be problematic as well. So let's go back. So the disadvantage, I said, it is hard to compare polygons that could be similar in size but different in shape. The example is both cnidarians and livestock have the same biomass, which is 0.1 GTC, but it will be hard to tell due to their different shapes. So that is very important. And then next here we see they say the same study also determined the distribution of biomass in 3 different environments. They did marine, deep underground, and land. The Voronoi diagram shows the distribution of biomass in each environment. The graph shows the proportion of biomass in different groups of organisms. So here we can see there are organisms on land, the organisms deep underground, and then those in marine. And again, since this is a Voronoi diagram, we can see land has the greatest number of organisms or greatest biomass, and then deep ocean or deep underground, and then we go to the marine. Let's continue down here. We can see that all plants live on land. Some fungi, actually most fungi live on land, while very few live in marine. Protists, we have some on land, but some are in marine. The animals, we have some on land, most in marine. Bacteria, most of them are deep underground, and archaea, most are deep underground. So we can see this is fraction of biomass, as well as the different organisms we are looking at. So I'm going to move to the next page. Explain the distribution of biomass in these 3 environments. Use the information in the Voronoi diagram and the graph to support your answer. So I want to take you back to the Voronoi diagram so that we can look at it carefully. We can see most of the biomass is actually on land, and then the next is in the deep underground, and then we have some in marine. When we look at the graph here, we can see most plants are found on land. Actually, it shows that all plants are found on land. There is nothing for the deep sea or marine. And then the fungi, we can see highest percentage of the fungi is on land. A little bit is going to be in the marine. For the protists, we can see that most of them are actually found in marine, while some of them are also found on land. We can see for the animals, the highest percentage is in marine. Bacteria, most of them are deep underground. And then for archaea, most of them are deep underground as well. So let's continue. So here I said the Voronoi diagram shows that the most biomass is found on land, and this is because the land provides better conditions like sunlight to sustain the organisms. All plants are found on land because they need adequate sunlight for photosynthesis, and because of this, marine environments would contain a lot of salty water that could inhibit growth, because water would leave the plants by osmosis to the surrounding salty water that could basically osmosis would occur, leading to the plants to dry out. And then we observe from the data that fungi are found predominantly on land, and this is because of the way they feed, and also they need sufficient gas exchange for respiration. Next, I went to animals. Animals are found on land and in marine environments. They are greatly adapted for gaseous exchange. As you can see, animals have different adaptations. Those found on land have lungs. Those found in the ocean or in the water have some gills and other ways in which they can get the oxygen into their bodies. So these great adaptations for gaseous exchange and feeding in the different habitats enable them to survive in different environments. They are not found deep underground because there is no food and the pressure is too high. Remember, the deeper you go, the higher the pressure of the water, so the pressure will be too high for these animals to survive. Now, the protists are found on both land and in marine environments because some can photosynthesize, needing light, and some can feed on phytoplanktons in the sea. That will enable them to survive in those conditions. So this brings us to the end of question 8 and the end to this video. Please do not forget to subscribe to our channel and please stay with us. I will keep sending you nice videos that can help you with your revision. See you next time. Bye-bye.